Welcome to our channel. If you have not yet subscribed to our channel, subscribe to our channel, click on the bell button to get notified about latest videos. Today in this video we are going to discuss about the bacterial cell wall. So first of all, what is a cell wall? A cell wall is nothing but a rigid layer made up of polysaccharide present outside the plasma membrane in cells of organisms such as plants, fungi and bacteria. We know that the plant cell wall is made up of cellulose, and in case of fungi it is made up of chitin. And in this video we are going to discuss a lot about the bacterial cell wall, the structure of the bacterial cell wall, the types of bacterial cell wall based on the types of bacterium, and also about the synthesis of bacterial cell wall and function of bacterial cell wall. So let's get into the video. First of all, let us discuss about the types of bacteria. If you see, the bacteria is broadly classified into two major groups, that is eubacteria and archaebacteria. What is a eubacteria? Eubacteria is nothing but typically a true bacteria with a peptidoglycan made cell wall, a nucleoid and a flagella for motility. That is, it has all the basic organizations of a bacteria. But if you see archaebacteria, it is similar in size and simplicity to that of the eubacteria, but it differs only in the molecular organization, and it is also an evolutionary intermediate present in between the eubacteria and eukaryotic cell. That is, from eubacteria archaebacteria evolved, and from archaebacteria only the first eukaryotic cell is found to be evolved. So that's all about the archaebacteria. If you see, the eubacteria is further divided into two groups, that is gram positive bacteria and gram negative bacteria, based on the gram staining process. If you see, the bacteria which is present in purple blue color after the gram staining process is called as a gram positive bacteria, and the bacteria which is present in red color after the gram staining process is called as a gram negative bacteria. And if you want to know more about the gram staining process, you can take a look at our previous video. The link of the video will be given in the description below. So that's all about the types of bacteria: eubacteria, archaebacteria, gram positive and gram negative bacteria. Now let us discuss about the structures of different types of cell wall. Let us start with the eubacterial cell wall. If you see, the eubacterial cell wall is made up of subunits called as peptidoglycan. So what is a peptidoglycan? The name itself says that it is made up of peptides and glycan molecule. The glycan part of the peptidoglycan molecule is made up of N-acetyluramic acid and N-acetylglucosamine joined together by 1,4 beta glycosidic linkage, and the peptide part of the peptidoglycan molecule is the tetrapeptide chain attached to the carboxyl group of the N-acetyluramic acid molecule. This tetrapeptide chain contains 4 different amino acid molecules. These amino acid molecules are L-alanine, D-glutamine, diaminopimelic acid molecule. In most cases, this diaminopimelic acid molecule will be replaced with L-lysine molecule, and finally we have the D-alanine amino acid molecule. So that's all about the basic structure of a peptidoglycan molecule. Now let us discuss about the cell wall of gram positive and gram negative bacteria. If you see, in case of the gram positive bacteria, the cell wall is very thick, that is about 20 to 80 nanometer thick, and in case of the gram negative bacteria, it is thin, that is it is only 2 to 7 nanometers thick. This is a major difference present between the gram positive and gram negative cell wall. Next, if you see the organization of the gram positive and gram negative cell wall, the gram positive cell wall has peptide interbridges which joins the peptidoglycan subunits together, and in case of the gram negative bacteria, this peptide interbridge is replaced with just a cross link. So if you see the image shown in the screen, you can clearly understand this concept. If you see, in case of the gram positive bacteria, the D-alanine molecule of the peptidoglycan subunit is attached to the diaminopimelic acid molecule or the L-lysine molecule present in the consequent peptidoglycan molecule, and this link is done with the help of peptide interbridge which is made up of glycine amino acids, and in case of gram-negative bacteria, this peptide interbase is replaced with just a link present between D-alanine and the diaminopimulic acid molecule or the L-lysine molecule. An important thing is that the carboxyl group of the D-alanine molecule is only linked with the consequent peptidoglycan molecules, and a mesh-like cell wall is formed which is very porous in nature and allows many molecules to enter the periplasmic space. And next, let us discuss about what is a periplasmic space. So before discussing about the periplasmic space, we must know what is a cell envelope. The cell envelope constitutes everything which is present above the plasma membrane. That is, in case of the gram-positive bacteria, it is made up of the plasma membrane and the cell wall, and the space present between the cell wall and the plasma membrane is called as the periplasmic space in case of the gram-positive bacteria. And if you see in case of the gram-negative bacteria, the periplasmic space is present in between the outer membrane and the inner membrane, that is the plasma membrane. So in case of the gram-positive bacteria, it is very much small compared to the gram-negative bacteria. In case of gram-negative bacteria, the periplasmic space is very large, that is, it accounts about 30 to 40% of the entire cell size. And an important thing is that in case of the gram-positive bacteria, the periplasmic space contains only some proteins and exoenzyme. Exoenzyme is nothing but the enzyme which is produced inside the cell and which acts outside the cell. So that's all about the periplasmic space of the gram-positive bacteria. But if you see in case of the gram-negative bacteria, the periplasmic space contains many types of proteins. For example, the channel proteins, the transport proteins. It helps in the conversion of nitrate to nitrogen gas in case of the nitrifying bacteria, and also helps in the energy conservation process of some chemolithotropic nutrition based bacteria, and it also contains some electron transport carriers in the periplasmic space. So that's all about the periplasmic space of the gram-negative bacteria. And an important and final thing is that the gram-positive bacteria contains teichoic acid in the cell wall, but it is absent in case of the gram-negative bacteria. Instead, the gram-negative bacteria contains a lipopolysaccharide molecules attached to the outer membrane of the gram-negative bacteria. So that's all about the difference between the gram-negative and gram-positive cell wall. Next, let us discuss about the archibacterial cell wall. If you see in case of archibacteria, the cell wall is made up of ethylenopolysaccharides. For example, pseudomurin and chondroitin sulfate. So what is a pseudomurin? The pseudomurin is structurally similar to that of the peptidoglycan molecule, but the only difference between the pseudomurin and peptidoglycan molecule is that in case of pseudomurin, this N-acetylmuramic acid molecule is replaced with N-acetyl telosomunironic acid molecule, and this pseudomurin is present in the cell wall of methanogens, that is methane releasing bacteria. And next we have the chondroitin sulfate heteropolysaccharide. This chondroitin sulfate is made up of glucuronic acid and N-acetyl galactosamine. This N-acetyl galactosamine is sulfated, so only this chondroitin sulfate is called. So it is found abundant in the extracellular matrix of the human cartilages, and this chondroitin sulfate is found in the cell wall of Allococcus bacteria, that is allophiles, which live in extreme salt conditions. And next we have the protein and glycoprotein made cell wall. These two types of cell wall are present in the thermophilous bacteria, which live in extreme temperature conditions. So that's all about the archibacterial cell wall. Now let us discuss about the functions of a bacterial cell wall. It prevents the cell from osmotic lysis. If you see, whenever the bacterial cell enters into the host environment, because of the hypotonic solution nature of the host environment, the bacterial cell will swell in its size, but it will not burst. The bursting of the bacterial cell is prevented by the bacterial cell wall, and next it is responsible for the resistance to the host defense system. It provides resistance against the host defense system. Sometimes it even hides the bacteria from the host defense systems, and it's responsible for the shape and integrity of the bacterial cells, and also it is responsible for the virulence of the bacterial cell, that is, it provides the pathogenic nature to some bacterial cells. Finally, let us discuss about the synthesis of the bacterial cell wall. If you see, it is also starting from the important metabolic substrate, that is glucose. From glucose, it is converted to glucose 6-phosphate, then to fructose 6-phosphate, then to glucosamine 1-phosphate, and then to N-acetyl glucosamine 1-phosphate, and it is converted further to uridine diphosphate N-acetyl glucosamine molecule. In some cases, this uridine diphosphate N-acetyl glucosamine molecule will be converted to uridine diphosphate N-acetyl muramic acid molecule, and also tetrapeptide chains will be attached to this UDP-NAM molecule, and all these small molecules will be brought together to form a peptidoglycan structure. These peptidoglycans molecules will be brought together via peptide interbridge and cross links present in the bacteria, and the mesh like porous cell wall is completely formed. So this is the entire summary and details about the bacterial cell wall. Thanks for watching. Stay tuned for our next video and don't forget to subscribe to our channel.